So you guys all have a sheet in front of you. There's a part of the lab where we have to wait for 10 minutes. So what I'm gonna do is get us started. And then during that 10 minutes, kind of talk about it a little bit more, okay? So we can keep moving along and not waste any of our time. So we have some materials here on the cart. When you're finished, they need to go back on the cart. Everybody will get one of these, okay? They are, when they say the lid tube, that's what this is. Did they put? We've got Gatorade, which you'll have a little bit of that. You'll swish it around in your mouth for about a minute. And you're going to spit it back in the cup. I know, lovely. We have ice cold, 99% isopropyl alcohol. You guys probably know that that's not to be drank either. What else? These are pipettes. They asked to be using a pipette. So the first thing I'm going to do is I'm going to pass out little Dixie cups and pour a little bit of Gatorade in there. If you don't want to participate, just say no thank you. And then it says to take the Gatorade and swish it around your mouth for 1 minute. Do you guys want to be timed as a group or do you want to time it on your own? As a group. All right, so when I pour it into your cup, don't do anything with it quite yet. Would you like to?
Yes.
Would you guys like to participate? Good. Yes. All right. What we're going to do is kind of swish it around in your mouth as if you're having mouthwash. We're going to do it for a minute. Okay, we'll do it as a group. Obviously, if you can't make it a whole minute, just spit it back into that little Dixie cup. So if you're following along on the sheet of paper for the methods section, it says pour about 5 milliliters of Gatorade into a small paper cup. That's what I just did. Do not swallow it and take the Gatorade and swish it around your mouth for about a minute. Is everybody ready? I will time you. All right, ready, set, go. Once you're done swishing it in your mouth for a minute, we will then carefully transfer the contents back into the cup. So what you guys are doing right now is we're trying to extract cheek cells. There's some salt and sugar in Gatorade. The salt specifically, the electrolytes in this Gatorade, are going to be taking those cells off of your cheek cells. We've gone for about 30 seconds. $15 seconds left. 10. And time. You can put it back into the little cup. Lovely, yes. So now I'm passing out a tube that has a lid on it. We're going to put the contents into Okay. It says to place your tube in an upright test tube rack, but they did not give me test tube racks. Okay, I guess. I can check what it is. Let's see if there's something here. Okay. So you're going to pour that Gatorade cheek cell solution into that tube with the blue cap. And then we're going to place them upright into this container. If it won't all fit, that's fine. Only pour about 5 milliliters if you can. So just dump the rest back. So 5 milliliters, if you look on the side, is like halfway up, or a little bit less than halfway up. You only need about 5 milliliters. I gave you guys some extra Gatorade. You're welcome. So then once you have it into that tube, you'll have to share. I put some of the test tube racks throughout the classroom. There's 4. You're going to place the tube into the test tube rack. Remember the location. If you'd like to label it, you can. If you're worried about forgetting about where it's located, there's some Sharpies up here. You can label it if you'd like. Or just remember where you put it. Maybe 4 corners or... All right, so we have about 10 minutes. It says to place the tube upright in the test tube rack and let it stand undisturbed for 10 minutes. You should begin to see cells collect at the bottom of the tube. So we're just trying to not bump these tubes. It says do not shake. After the 10 minutes up, we're going to pipette about 2 milliliters of soap solution. And these are different soap solutions, so I'll pass those out in a little bit. And then after the soap solution, we're going to let it sit for 5 minutes. Then we're going to add some isopropyl alcohol that's cold, it's on ice. Then we'll be able to start seeing the DNA. Okay. So while I walk around and pass these out, so we have DNA in our body. Does anybody know where your DNA is located? Or you can take a guess.
Nucleus.
Nucleus, yeah. So your DNA is located in your nucleus. We're also pretty lucky. We have DNA in our mitochondria as well. DNA is found in your chromosomes, which we talk a little bit about chromosomes in lecture on Monday. And the chromosomes are made up of DNA and protein. The protein is called chromatin. They are tightly wound together so that we don't damage our DNA. They say that if you were to pull and untangle all of your DNA out into one long line, be about 6 feet tall. Anybody 6 feet tall in here? Yeah. So your DNA is about as tall as you if you're 6 feet tall, or know somebody who's 6 feet tall. And that's pretty tall if you think about how small DNA actually is. So you're telling us that we have a cell. Okay, we've got a nucleus, and we've kind of got some mitochondria too. But for the purpose of today, we're just going to talk about the nucleus. Okay, so there's your nucleus of the cell. You've got a plasma membrane. We've talked about that before. Does anybody remember what the plasma membrane is made up of? Yeah, phospholipid bilayer. So we've got these phospholipids with fatty acid tails. Everybody kind of remember this? I'm just going to roughly sketch them. Okay, so these are considered fatty acid tails. This is called a phospholipid bilayer. Okay, inside our nucleus we have chromosomes, which kind of look like this. Or maybe you've seen them before. They come in pairs because, well, for humans anyways, we are diploid, which means that we're 2N, which means we have 2 pairs of our chromosomes. Our sex cells are 1N, so that when a sperm cell and an egg cell come together, they're both 1N, they make 2N. So now we get pairs like this. The chromosomes are tightly wound DNA structures. So what I like to do to kind of show that, and there's a picture like this in your book as well. Okay. So this is your DNA. It's that double helix shape, right? You've got, so this is a double helix. It's made up of a sugar phosphate backbone. Okay, and then these are nucleic acids or nucleotides that make up the prongs of the ladder. Your DNA is tightly wound together into chromosomes using a protein called chromatin. Okay, we'll talk about this in lecture on Friday. So think about it. Your DNA, it's very small. It codes for different traits and functions. DNA encodes for proteins, and the proteins allow for functions, things as simple as movement and skin color, hair color, all of these different functions, breathing, that goes down to some very small patterns of nucleotides. It's tightly wound into the chromosomes, and the chromosomes are found in your nucleus. So we've got about 4 minutes left here. I think of it as if I were to throw, I always use this napkin as an example. Okay, so here's a napkin, right? You can also use a piece of paper. If I were to throw this napkin like this, how far do you think that I can get this napkin? Just like this. It's not too far, right? Let's try it. Cool. Okay, so it didn't go too far. Kind of damaged the napkin here, folded it a little bit. It got damaged. Okay. So now if I tightly wind up this napkin into a small structure, how far can I throw this napkin? Oh yeah, I'm pretty athletic. Look at this. Pretty far, right? So the same thing goes for your DNA. So it's tightly wound into your chromosomes. So one, it doesn't get damaged. Can you imagine if our DNA was just kind of like floating around, like untangled and just kind of sitting there? It could probably get easily damaged. So the protein, chromatin, and having it tightly wound up on itself allows for the protection. So that's what that sugar phosphate backbone is. It doesn't allow for the damage. It also allows for it to travel. So when there's cellular respiration or mitosis and meiosis, cellular division to make more cells, it's not like our DNA is just floating around and getting damaged, because you don't want that, right? That's what is important about that structure. I want to see what other questions. So it says, where is DNA found? What is a chromosome? This is a chromosome. What's it composed of? DNA and chromatin, a protein. What is the size of a DNA molecule? The size of a DNA molecule. You don't need to know specific of that. How are you able to visualize your DNA? Okay, so what we're doing today is trying to extract our DNA, which is inside the nucleus. The first thing we did was extract some of it off your cheek cells, right? We got some of the DNA off your cheek cells. Some people have maybe done it in another science class where you use a toothpick and kind of swirl it up against your cheek, okay? We use some sort of physical force to get the cheek cells off our cheek, okay? Now we're just kind of letting it sit. We've got about 2 minutes left until we do the next step. The next step is that we're adding soap. Okay, that seems silly, right? But we just talked about that this is made up of a phospholipid bilayer. Keyword being lipid. And it's the same reason why you can use Dawn dish soap and wash the grease off your pans at your house or your apartment or whatever, or why you wash your hands to get gunk off your hands. With soap being a base, it can essentially break up lipids, the fat, the grease. That's kind of how I think about it. So the soap that we're adding is going to essentially break open the phospholipid bilayer. There's also a phospholipid bilayer or a bilayer around the nucleus, but for the purpose of today, just think of that the soap is breaking this phospholipid bilayer because here's that plasma membrane. We want to get to the DNA that's in the nucleus. Okay?
What did you draw underneath the word nucleus?
Nucleus. Those are chromosomes.
Where
Yeah.
are those at?
What's that?
Where are those?
Those are found in our nucleus. So that you have 23 pairs of chromosomes. I'm not going to draw all of them, but you have 23 pairs, which maybe you've heard of before. 22 of them are autosomal, and one pair is your sex chromosomes, which determine your sex. And those are either you have an XX or an XY, which we talked about, determining male or female. So these are the chromosomes. The chromosomes are made up of tightly wound DNA. Okay. So why are we using the detergent, the soap? Is to break open that outside of that cell so we can get to the nucleus where the chromosomes are that house our DNA. Because we want to look at DNA today. Why is it important for science to study DNA? The first couple of paragraphs there kind of talk about it, but DNA can be used for a lot of different things, especially like criminology. Oh, good. And we'll talk about that in the next 5 minutes. So right now you guys have some soap solution and you have pipettes. Remember how to use the pipette? You squeeze the top when you're outside of the liquid, put it into the liquid, release, and put it into the solution. It says to pipette 2 milliliters of soap solution. So what you should do is if you have 5 milliliters in there right now, you should put soap solution up until it says about 7. You don't have to be perfect here, but as close as possible. And then it says do not shape, but you can gently invert 5 times. What invert means is you're going to put the cap back onto the tube and kind of go up and down, up and down. Do that 5 times. Again, looks like this with the soap solution. That's invert, invert. Don't shake it. You kind of go slow. Do that about 5 times. Once you do that, you can place it back in the test tube wrap for about 5 minutes. Once everybody's seated again, I'll start that timer so I know that we're all on the same page. We wanted to mix the soap solution in there, but we didn't really want to create a bunch of bubbles, right? That's why we didn't like shake it. We just inverted it. We wanted to mix it together, but not create a lot of bubbles, which everybody looks like they did a good job. Perfect. So I just started a timer for 5 minutes. So the reason studying DNA and being able to see DNA and why it's important is that your DNA is specific to you. Even if you're a twin, even if you're a sibling, you as an individual have specific DNA. So think about a crime scene. If they can get a sample of DNA, they can match it to the crime scene or things like that. That's super important. I worked for my master's thesis on identifying mitochondrial DNA and understanding every single nucleotide that's in a DNA sequence of an individual organism. Okay, so other things are for genetic testing. Nowadays, you can also find out the gender of a baby, like I think it's after 9 weeks or something like that, through a blood test. The blood test, they look at the individual's DNA and some of the hormones and things like that in the blood. But some of those specific things that are specific to the individual because of the DNA. So the DNA, you have those patterns of the A's and the G's, the T's and the C's, and how they match up, and we talked a little bit about that before. And that pattern is specific to the individual, which is very interesting. So that's kind of a couple of reasons why we study DNA. Like it says on the first page, it's your genetic fingerprint, it codes of life. It says the length of a DNA per cell is about 100,000 times as long as the cell itself. However, DNA only takes up about 10% of the cell's volume. Individual chromosomes can be studied using microscopes, and we're going to look at a couple of pictures of chromosomes in lecture, so you guys will be able to see that a little bit better. But the double helix of a chromosome is so thin that it can only be detected through high-tech procedures. Chromosomal DNA from a single cell is not visible to the naked eye. However, when chromosomal DNA is extracted from multiple cells, the amassment of it, or the quantity of it, is what we can see. So that's why we can't see DNA to the naked eye, but we have a bunch of cheek cells that we extracted from your cheek and that are in that solution. So although we can't see DNA of a single cell, when we put a whole bunch of copies of the same thing over and over and over, we can see it a little bit better. Another tool, and we'll talk about this in lecture as well, is called a polymerase chain reaction, a PCR. And this is essentially a copy machine of DNA. So I think about it as if you have one piece of paper that's so tiny you can't see it with the naked eye, but you go to the copy machine and you make a whole bunch of copies and millions of copies of that piece of paper and stack it on top of itself, you'll be able to see it eventually. So that's kind of what that PCR does. And that's, we're not using a machine here, but we've got lots of copies of the same thing stacked on top of each other, so you'll be able to see that a little bit better. So we've got about 1:50 left. Let's see here. We talked about the detergent. It breaks open the cells by destroying that fatty membrane that encloses them. This releases the cell's contents, including the DNA, into the solution. The next thing we're going to be using is ethanol. And this causes the gases dissolved in the water to be released, which you may observe some bubbles. But this also allows for us to precipitate the DNA. So we're kind of drying it out in a sense. So when you guys use hand sanitizer on your hands and it has alcohol in it, you start to get like dry hands, right? Or you wipe a table with some Clorox wipes that probably have some alcohol in it, and it dries a lot faster than if you were to just spill some water on the table. So alcohol has some properties of it are that it dries things up. And that's how I kind of remember that the alcohol is what's going to help us kind of precipitate the DNA. The word precipitate means to make a solid. So we're going to make the solution into a solid. I'm just going to kind of come around and look and see what. Oh, yeah. You started to have some DNA in there. Can you kind of see? That's good. All right. We're getting close here. We're going to be adding the ice cold isopropyl alcohol in about 20 seconds. So what I'd like you to do is I'll pass these out, but when you're done with them, if you could please come put them back on the ice so that they're cold for the next group. So we just share, I'll get you guys new pipettes. You know, normal alcohol is like 4, 5, 6, 7%. This is 99. You can do the math. Don't drink this stuff.
It's all the way up.
It says to about 13 or 12. Is that right?
Yep.
12 or 13. Good. And then let me get you guys some new iPads. Let's have. How many do I have left? 2, 4, 16. For the alcohol, can you guys share that
Yeah.
pipette? For the alcohol, share this pipette. For the alcohol, share that pipette. Go ahead and add the isopropyl alcohol. Share the pipette with your table partner. And then when you're finished with the container of the isopropyl alcohol, just come put it back on the ice, please. The properties, when it's cold, it allows it to be more active. So it's just 12 to 13? Yep, 12 to 13. So just fill it up to the 12 or 13. Until the total volume is about 12 to 13. You should then start to see two distinct layers, the Gatorade and the alcohol. Be careful not to shake or mix the tube. You'll break the DNA and it will go back into the solution. We want to see that solid in the middle. We're going to let it says to sit for 10 minutes, but we'll just do what we can. It says a white filmy layer will appear between the Gatorade and the alcohol. This is your DNA. DNA yields will vary by the student within class. It'll also vary if you just had lunch or something or just had a snack or a drink, you'll have more or less DNA depending on that. So it says on the lab, it's like, do not eat a drink or drink an hour before lab. I don't ever enforce that because most of you come from lunch, so I'm not going to be like, don't eat before coming to class, especially now. Don't you guys have like specific times you have to go or something? Is that not true? I don't know. Perfect. So we're letting that sit. You guys maybe we'll be able to start seeing some precipitated DNA. I also brought treats. One treat. So, like a month ago was my birthday, and some of you guys were quarantined on a Wednesday when I brought snacks. So this is me bringing it and making it available again. So there's some veggie straws and candy, but don't eat them in lab, but you can take one to go or down one. While we're waiting, if you want some Sour Patch Kids, which are my favorite. Around, and if you could put, let's just leave the soap on the tables because then I'll pass it back up. Does anybody have any questions over anything I talked about? It's okay if we're not getting this the first time around. We talked a lot more about this on Friday. Oh, that's There will be a lab quiz. It's on my Wartburg. It's due at 11:55 p.m. tonight, and you have 45 minutes to do that. So whether you do it right after class or 7 p.m., doesn't matter to me. We're doing chapter 22 tomorrow, or not tomorrow, I keep saying tomorrow, on Friday, which is DNA and DNA technologies. Your chapter 6 lecture assignment is due Friday. Most of you have it done though, which is good. Good. I'm going to kind of walk around and see what we've got. Oh, yeah. Is this one yours, Nick? take it. This one's pretty good. So we're just kind of letting that alcohol and the Gatorade settle. Eventually we'll be able to see two distinct layers. And we're about 5 minutes in of waiting of our 10. Biology is a hurry up and wait kind of science. Hurry up to do something and then you wait to see what happens. About 2 minutes left. You should start to see two layers, a Gatorade layer and an alcohol layer. And then in between those two layers, or maybe kind of floating within, those like chunky, like saliva looking things. That's the DNA. Now, some will be better than others, and that's just kind of how it goes, especially if you just ate. You may not see a lot in there. It's also because your mouth was digesting food, eating, swallowing the food, so the cheek cells that would be easily accessible to be extracted wouldn't be right there. And then once we do see some DNA, you don't have to, but we have some of these little itty bitty micro or like PCR tubes if you'd like to take your DNA home with you. We also have some yarn if you'd like to make it into a necklace and wear it around. Or we like to look on what I did today. You can do that too. You can use, if you'd like to do that, I know it's super fun. So you grab one of these tubes and then just use one of these skewers to just kind of pull the DNA out. You can put it in that tube. I'll lay that here. No pressure. If you just want to throw it away, too, that's fine. I won't be feeling from here. Oh, yeah. You're kind of good.
Where is it over here? Over there.
A couple more minutes and we'll let them sit. I was going to play music while we're waiting for our DNA. But I want the speaker to be here. Really, this is beyond my expertise. I've done this before. I looked at your guys' responses and country won by a landslide from that get to know you thing at the beginning. Man, I'm gonna be able to maybe 7. Okay. Alright. Oh. Maybe this. Oh yeah. Well, that was the 10 minutes. If you'd like to take your DNA with you and show your friends what you did in class today, you can. Other than that, the cleanup is you dump the liquid down the drain of the skinnier tubes, leave the soap Leave the pipettes on your table. Wipe down your table. The skinny tubes that had your Gatorade and spit in it can go in the trash. Your Dixie cups can go in the trash. Wipe your tables down as I try and figure out how to do this. Yes. Yeah, you guys can put the pipettes away too, actually. I was gonna have you keep them, but then I was like, COVID. I'll get new ones.
Here.
I've done it. Thanks. Yeah, enjoy. You guys love the cookies. There's a whole thing of cookies in.
Celebrate a title. The NBA Finals on ABC. Rise together.